So we learned earlier that this particular species of
ROV
coral,
bridge, move complete.
unlike many of the corals,
Copy
is
that bridge,
able
thank
to
you.
root itself in the
That last
soft
move was just
sediment.
completed.
It has
You
a
guys
more
happy with
extensive
continuing
rooting
going on?
structure or
think so,
attachment
yeah.
structure
About 30
than
meters,
most
same
corals,
move.
so it
Yeah,
does not
sounds
require a hard substrate under this layer of soft sediment that we're seeing.
Bridge,
It actually
ROV
can
nav.
attach itself
Go
or
ahead, RV now.
root
We'd like
itself
to make another
within
move,
the
exactly
soft
the same
sediment.
thing. 30 meters at 325 degrees, speed of 0.2, if that works for you.
30 meters, range 325, speed decimal 2 works for me.
Excellent. Thank you.
Copy that. You can come wide then.
Hit it with lasers yet?
So we'll be
I think
moving
we
on
landed a bit.
here and hoping that we get to a place with rocks.
run
It
the
sounds like the
lasers.
geologists are getting kind of really excited about that aspect. And
ROV now bridge, move initiated.
Well, we know
Good
that when
copy,
we
Bridge,
find
thank you.
rocks, we're going to find some great organisms. So don't get
Right,
us
lasers
wrong.
are
We love
on.
rocks with critters.
Two of them.
And Les Watling pointed out that every one of the Acanella bamboo corals should have a shrimp on it, and he thinks that the one that we were just looking at was an immature shrimp. You have more burrows.
We promise we have not just been going in circles and we see the same one over and over. They all look so similar and they have just one shrimp.
And here's another one on
Oh.
the left.
Might call us wrong, though I don't see a shrimp with that one.
I
but-
think the shrimp was on the sediment,
Oh, good.
so
good.
I think it was there.
He was out mowing the lawn or something. Yeah, it's great.
There it is on the
Oh,
right.
yeah, there he is. Maybe that was the one who we saw a few minutes ago.
Right. Pushing forward a little.
And we're seeing lots of these grazing paths of the heart urchins. Earlier we came across the the shell remained. There was no living organism of a heart urchin, so it kind of verified, or potentially verified, our Scott France's theory that these were likely to be heart urchin burrows. And the abundance of them is indicating to us that there's food in this region, and there's certainly plenty of life here. We always think of the sediment plains as being low areas, or areas of low diversity or low abundance of organisms, but maybe they're just hidden. There's plenty of life within the sediments, and we are seeing several organisms living on the surface, of course. Not as many fish as we might see in other environments. If you follow us throughout our next 18 or 17 days of dives, we are going to be visiting completely different areas, many different depths, as deep as over 3 kilometers and as shallow as maybe, 300 meters? Yeah, so we're going to see such variety of environments. So we're really excited about that.
And this is an environment that I haven't been exposed to very often. I'm more used to diving in shallower areas, so it's really fun to go someplace so different that you've never seen.
So we haven't seen any kind of ID, as far as I can tell from the chat room, for at least the genus of these large brittle stars. We caught one swimming earlier in the dive. I'm sure that will be on the highlight video because it was pretty amazing. Is there anyone out there who can help us with the brittle star? I know it's in the class Ophiuroidea. Is that correct? Trying. I'm going to have to learn some of these.
Leslie, you know an impressive amount of information about the biology for a geologist.
Oh,
so
that
we
is
just say.
the
Sure.
kindest thing you could ever say. Well, I will admit that I've been on submersible
do
dives
the
where I
board
was the
top
only
screen.
geologist, so I had
Yeah,
to
go
learn,
for it.
and a lot of
Smooth it in.
biologists, and I learned a lot, especially echinoderms. I'm
let's
quite
let it go.
fascinated with them, I will admit. But I can't call them by the species name.
That looked like there was something gelatinous in the water column that just floated past us. May have been a salp. It was kind of hard to tell because if you noticed it, it was almost 100% transparent. So it's hard to identify.
And we're at 1,715 meters now, so we've come up, about 40, 45 meters since our initial place that we landed. Didn't we land at 1760?
I thought it was 1720, but I'm not positive on that.
This is nav. It was 1736.
So yeah, we landed. The initial depth that we first landed on was 1,730 meters.
Thank you, Nav.
And now we're at about 1,703 meters.
Oh, this one. That's Sirius.
Oh, okay.
So for folks that are joining us from home, we are conducting a science AMA on Reddit today. And one of the questions that we got was, how would you recommend people get started in this field if it's the first time that you're ever trying to get involved with deep sea exploration? So for all of our science leads on the ship and folks at home, our shore team participating, why don't you guys answer that? That might be a good way to learn a little bit more about your backgrounds and how you got started in this field.
Oh, check out this hermit crab carrying an anemone. So, to answer Casey's question, I was interested in marine biology since a young girl, and my parents tried to talk me out of majoring that in undergrad, but they couldn't. And so then I've proceeded to get a PhD at Florida State University, and now have been at the USGS for 16 years. And I'm very fortunate that I get to do things like this, and it's the highlight of my year all the time, every year when I get to participate in these types of expeditions.
Oh, and there he goes.
Zimbabwe bass.
Maybe we could get a zoom. Oh, well, not next to the skid. Ken Sulak has let us know that the brittle stars are likely Ophiomuseum limani.
Tight to one of these large ones.
So it would be worth zooming in on one of these.
I was looking at that too. I guess we. Well, actually, do you want to zoom on a brittle star real quick?
Yes, that'd be great.
Yeah,
Please.
do that. Get closer, video.
It looks like that ship move just stopped, so you guys have plenty of time.
Great, thanks, Nav. Video, if you want to go in.
So again, the genus Ophiomuseum.
ROV nav bridge, move complete.
Copy that, bridge. Thank you.
They're beautiful. So as you might imagine, these are relatives of the sea stars. Very common. I've seen them everywhere that I've ever been. And they're feeding from underneath the plate.
Video's clear.
All right, go ahead and come wide, video.
Sadly, we can't flip it over so we can look at the mouth, because I know that's where we get the identification down. We're going to take a look at this feature here that sadly might be marine debris. Not sure.
The
Yeah.
chair.
Yeah, this might be our first
Good.
seafloor.
Back end of a
looks
surfboard.
related
You know, the leash.
to diving, maybe?
Oh yeah. Nav just said that it might be a surfboard, and that's the leash that you strap to your
that
ankle.
Coming on it,
definitely looks
video.
like a leash.
Oh, it's a buggy board. Yeah.
Oh yeah.
It's got words on it.
But that's more of a pad.
Oh yeah, no,
Huh.
that's
Yeah, it's a
electronic.
cable.
Velcro.
Yeah. Yeah, I was thinking
oh yeah,
diving.
a dive computer maybe.
And then like the BCD like kind of opened up. I don't know, it's kind of hard to tell when it's
Yeah,
partly-
Rachel Bassett agrees, BCD.
Yeah, the Velcro's there. We could see what it was.
Yeah.
Just look up at the where the connection's made.
So Ken and Tina, what did you think about the Brittle Star? Did you think it was the
Words on
Opium
the
Museum?
hose.
I guess you'd already determined that, but
Yeah.
were you able to
C1 hose.
pin down a species from that view?
24. Here you go.
R237
We have some pretty good cameras here.
890.
That's amazing.
.24 gauge.
And of course, in the background, the sediments are telling
300
us.
volts.
We have,
I see multiple species of pteropod
Yep.
shells laying
Yep.
on there.
yep,
I don't
and
think,
lots of echinoderm spines,
Yes.
which is no surprise.
I don't think this is BCD because it indicated
No.
voltage.
Yeah, is that just like a
Huh.
put on a chair? One of those
It's like a chair
massagers
cover.
or something?
It has straps for sure.
Oh, like one of
that,
those
like a,
massage chairs?
Yeah, it
like,
looks
or
like-
like a chair warmer or
It looks like a
something?
chair.
it's from a yacht, obviously. Oh well.
I would say that's definitely man-made debris.
Yep.
Yeah.
All right, video.
Clear,
Happy
full
with
wide.
that. All right, now we'll do a little push, and then we can get another move in.
Yeah, sounds good. We'll make another move, 30 meters. Same heading should put us right over new waypoint 2.
Perfect.
It's too far.
Stephanie, maybe you could tell us a little bit about how you got involved with
It's 45.
your studies of pteropods.
Oh, actually, so initially I worked on
Yeah,
deep
Nav,
sea
you can put that moving.
squids,
Copy that.
and I was
Bridge, this is
interested
ROV
that
Nav.
whole time I was working on
This is
them
bridge over.
Would like to make
for my
a
graduate
move
work
of
at the
30
University of California,
meters
Berkeley.
At 325
I was interested
degrees,
in,
a speed of 0.2
how
knots, please.
speciation
Understood.
of
30
different
meters,
taxa happens
325
over
degrees
time
at
in the open
decimal 2
ocean.
knots. Thank you.
Good copy,
But I
Bridge, thank
knew
you.
that asking those kinds of questions with squids was not going to be easy just because it's difficult to get the number of squids that you need in order to do those kind of studies. So I transitioned to studying pteropods because you can do plankton tows where you drag a net through the water, and you can get, if you get lucky, you can get the right number that you need for those kind of studies. So that's how I started working on pteropods, was to look at connectivity. So the idea of intermixing of individuals between different geographical
Is that a
locations.
model?
And
Need to load
one of the ways that
lasers?
we study that is to
ROV now
collect
loop initiated.
Copy that, Bridge,
specimens
thank you.
of what appears
Yeah, go for
physically, morphologically, to be the same species from
wood,
different
wood, yeah.
locations. And then we sequence the DNA and compare to see if actually
piece
what
of
we
wood, this
think
is
is the
gonna be a
same
shipwreck.
species
Cover up all
um,
this, yeah.
truly is. Like, do they have, is their DNA similar enough that we would
Sorry.
consider them the same species, or is their DNA different enough that would be
Different claw
that
there
we
on the
would
right,
consider them
lasers.
separate species,
The
but
dark one?
visually they look the same.
Let me get a little closer.
So
and
I
give
started
me
working on
one sec.
pteropods when I was a postdoctoral researcher at the Smithsonian Natural History Museum. So that looks
maybe it's an anemone,
it looks like an anemone, maybe a tube-dwelling anemone, a ceranthid, or it could be actually
flytrap anemone, I think, or maybe not.
Looks like there's a tube sticking out.
Yeah.
Any anemone experts out there would like to chime in on the chat room and see if this is actually. This one looks more like a ceranthid to me, but my expertise, as I've been talking about, is in water column dwelling animals, not benthic animals. So.
Video's clear.
All right, go ahead, come on.
Cheryl was talking earlier when we saw an anemone about these tube-dwelling anemones, so it looks very similar to a non-tube-dwelling anemone, but the tube that they secrete that anchors them within the soft sediment can actually go down quite a ways. So if you're ever out, it's not easy to collect one of these if you're interested in studying them. I know people that have been doing research on them, and they said it was very difficult to get the entire tube out of the sediment.
We actually did that on one of the first cruises that I went on in the Gulf of Mexico, and we used the suction sampler, and it was quite a long endeavor to try to get him out. But it was very impressive when we got him back to the ship to see how long that tube was. And
you were saying they could go down several feet sometimes, the tubes?
That one was at least 2 feet, but it might have been even more like 3 feet, about a meter.
And did the animal within the tube extend that far as well? Or do you think they grow as they grow, and so they're only occupying the shallowest part of the tube?
I actually don't recall how far down the animal went. I think it was shorter than the tube, but how much, I'm not sure.
Watch Lee, this is Nav. Wanted to let you know that we've moved another 100 meters, a total of 200.
So we've now reached another 100-meter transect that we've just passed. So total, we've gone 200 meters since we reached the seafloor.
We just passed another tripod fish. I think that's maybe the third one we've seen so far. So I think we passed another one when I was talking about pteropods. The genus of those tripod fish, bathymetry. I'm not sure if I'm pronouncing that correctly. But I don't know if the three that we've seen are different species or if they've all been the same.
Oh, no.
Do we have anyone in the chat room who'd like to give us some information that we haven't mentioned? Feel free to call in.
We're currently at 1,704 meters. So it's hard to tell because the Deep
ROV
Discoverer is staying level with the sediment, but we actually are moving upslope as we travel along here.
Nav, this is Watch 1.
Go ahead, Watch 1.
Are we actually on the scarp feature now? Would you be able to tell that?
Yeah, we're coming right up to it. It's a pretty shallow slope, so we've been on it for a little while.
Great, thank you.
No problem.
Watch leads. Any interest in item coming to center? Yep. All right, video. Give me 1 sec.
I think is that Umbalula?
That's what I was thinking. So
Alright,
that's
video.
a stocked sea pen.
Go ahead, come in.
Yep, Scott Francis agrees it's Umbalula. Wow, look at those beautiful polyps.
Those
That's
are beautiful.
Gorgeous.
Now, these were seen in the Gulf of Mexico as well, correct?
Yes.
Yeah.
I don't know if it's the same species, but...
maybe Scott can verify that. One of the goals is to understand more about connectivity. Maybe you could talk about that a little, Cheryl.
Stephanie
Or
just
Stephanie.
talked about that, but yeah.
I'm sorry, I was out.
Yeah, so in terrestrial systems, it's easier to think about connectivity because, for instance, if you're a stream organism, you're going to be moving only within that stream or neighboring streams. You're confined to a narrow set of places to travel. But when we look at the ocean, the barriers between places are nowhere near as obvious.
Bridge over.
So,
if I'd like to make another move, it will be the exact same thing. 30 meters at 325 degrees, a speed of 0.2 knots, please.
and it's
Video, do you want anything else?
each
Understood.
species
30
may
meters
have
at
their own
325
patterns
degrees,
on how they move, how
Good
far they
copy,
move.
Bridge, thanks.
Often in the juvenile
Sorry,
larval
Sylvia.
stage is the only time that they're able to move around.
Okay,
And
great.
once they
You can
settle
come
down,
wide then.
they're at that new spot for
Lasers
life.
on the
So,
base, and we will
determining
move
in a
on.
population how many individuals were always there versus those that have come in as larvae from other places is something that we try to do. The way that we really can do that now is with genetic fingerprinting. When you can get the family structure of a
ROV nav
population
bridge,
and
move
see which
initiated.
ones look like
Copy
they
that,
were
Bridge,
local
thank you.
versus those that came from a distance.
Yeah, copy that, wash leads. If you see any, just let me know.
Yes, Ken and Amanda, we have noted that we should be stopping a little longer to get some zooms on the fishes, and we will certainly do that. And I hope that didn't jinx us from seeing fishes.
So Cheryl Morrison of USGS was just talking about connectivity of organisms in terrestrial systems. One of the things we're interested in looking at possibly in this cruise, collecting the same types of organisms that we've collected in the Gulf of Mexico, and then taking subsamples from those specimens
Fish coming up.
and looking at DNA fingerprints from those and comparing those that we collected here off the eastern coast to those that we've collected in the Gulf of Mexico, and see if the populations are interconnected between those two systems.
All right, video, you want to go in on
Go ahead
fish?
now. Go ahead, watch. Yep, working on it.
Wow, we have a beautiful tripod fish here. They sit on their pectoral and caudal fins and wait for some lunch to come by.
As you can see, they spend a lot of their time sitting stock still, and as Cheryl said, they're waiting for food to come by. And one of the reasons that those lateral fin rays are so long and sticking out above the main body of the fish is so that they can detect water movement, and so they have sort of like an early warning signal that something that could be potentially interesting to eat is coming along, or it could be a warning that something potentially dangerous that would want to eat the fish is coming along. So then the fish has a sort of those fins extend further out from the main body of the fish, so it can detect from further distances things that it may or may not want to respond to.
Washley, is there any specific part of the fish you would like a zoom on? Or an area to focus? Or is this good? Alright, copy that. Alright, video, is there anything you want to do? Alright, I think we can come wide then.
Amanda Demopoulos pointed out that there's a notch right on the bottom side of the caudal fin. Yeah, I wonder if that's one of the features that would probably distinguish species, I imagine.
Another thing viewers probably noticed that the eyes of that fish are very small. Keep in mind that without the lights of the ROV, it is completely dark down here because we're at 1,698 meters. So other than biologically produced light or bioluminescence, there's not much that can be seen, which is again why animals down here have developed other means of sensing their surroundings besides relying on vision.
And this here looks like a Bathypathes black coral, if I'm not mistaken. Could we get his...
Yeah, copy that.
Could be a sea pen. Looks like a peninsula from a distance.
Oh, okay. It actually is most likely a sea pen.
Good, come in.
A very different looking one than we saw earlier.
It's a nice one.
Nice
big
lasers, please.
branches.
Nice.
Yeah, so this is definitely the genus Peninsula. Earlier we saw a sea pen, so they're both in the same group, the sea pens, the Peninsulaceae. But this is definitely a different genus, and you can see it has what are called leaves. So those things that look like side branches or pinnules coming off are fleshy extensions from that main part of the body, and the polyps come out of those fleshy extensions. So essentially you have one giant polyp with a lot of arms, and then a whole bunch of smaller polyps arising from those arms.
Great, thank you, Scott.
So again, this is a type of sea pen, and it's, as Scott was just explaining, it's one very large polyp with a bunch of smaller polyps branched off from the main polyp. And like many of the organisms down here, it is
whenever you're happy.
Capturing particulate material that's going by in the current as its food source.
To the right, we can do
Looking at you 80 degrees.
Alright, let's do a very, very quick zoom video. And then I have to book.
And here we have a different type of sea pen.
With the shrimp on it.
With the shrimp.
Alright, just give a few seconds video.
At a first glance, this looks a little bit like a Halipterus.
Mm-hmm.
Maybe not. It depends. So one of the ways that you identify sea pens, one of the first things you look at is to see, are the polyps coming directly out of the stem, or are they arising on ridges or leaves? And from what I can see, these do look like they're coming directly out of the stem, which would suggest something like Anthophyllum or Prototylum.
Looking back at you.
It almost looks like they're little pink
ROV
eggs
Nav Bridge, we have arrived at our
close
new
to the
video.
base of
Copy
the
that
polyps.
bridge. Thank you.
Oh, yeah.
Yeah, the eggs would be formed inside the gastrovascular cavities of basically the stomach, and they're hanging on little sheets that subdivide the stomach. So absolutely, I would expect to see those there. And I saw a small shrimp on the back of that colony.
Sorry, Watchley, we're falling behind Sirius. It's time to move forward. If we see another one, we can stop and get closer views of it. Did you copy, Watchley? Oh, we were falling behind our Sirius, so we had to move on and catch up. So we had to leave that specimen. It depends. So it's also take into account the physical zoom of the camera, but also the distance between the actual camera and the specimen. So it depends how close we are to it relative to the zoom as well. Yes, pretty much. Welcome. This looks a little different.
So going back to that tripod fish we saw, Ken Sulak has some interesting information that the notch is actually a subgenus character. Several species have a notch like that, but the function is not known. He said that the elongate pectoral fins are provided with thick nerves that go directly to large extravertebral ganglia, which is evidence of important sensory function. Very interesting. Thank you, Ken.
Copy.
And here we have another sea pen. I don't know if it's the Holyptera genus again,
It
or
looks similar to the last one, but yeah.
Yeah, I think I said Holypterus before we got a zoom on it. I believe these are something like Anthophyllum or Prototylum, but this is, yes, similar to, I think this is now at least the third one that we've seen. So similar to what we just saw a short time ago, where the polyps are arising directly from that main, what's called a rachis.
That was weird. I thought I lost footing or something.
Oh, and there's another
Acanella
Acanella
right in front. Yeah.
All right. Do you want to note that now?
Looks like maybe it has one or two shrimp.
Yeah, we'll do.
Yeah, 1549 complete columns with D2 was lost for about 2 seconds or a second.
Yeah, what is that?
And then everything was regained.
Sorry, but I'm just readjusting.
Bridge, this is Nav.
This is the bridge, over.
We just wanted to know how hard are is the DP working to hold this position right now?
Mostly it's just the bow thruster is working about 50 to 60% of its capacity. The stern thrusters and props are not working that much. So it seems to be somewhat balanced between the forces of the currents and the wind. We just get a little slap occasionally
down now,
from the wave action.
Gotcha.
Excellent, thank you very much. Yeah, the bridge said that the DP is not working terribly hard, so we should have plenty of power going down to the vehicle.
Yeah, I don't think we lost power to D2. I think something happened with the communication link.
There'll just be like an odd glitch. We weren't in like an awkward position or anything.
Not sure.
So it looks like this coral actually has 2 shrimp or maybe mysids in it. I guess shrimp. Though the ones we saw previously only had 1. This one's got a couple.
Do you want me to go ahead and make another move, or you're coming above him right there?
When we finish with this, I'll do a little push and we can keep moving.
Sounds good.
ROV nav bridge.
Go ahead, Bridge.
Based on your last statement, are you having power issues with the ROV right now?
No, we just noticed some communication failed for a couple seconds on our screen and then came back on, and I was just wondering if it had anything to do with working too hard with the thrusters, but I don't believe it is that.
Okay, we're just checking in since we heard that statement. We wanted to keep everyone up to date. Thank you.
Yep, no problem. It was just a thought.
We're going to zoom in on this little pink amphipod we saw swimming into the scene earlier.
Copy that.
We won't get a really tight zoom probably because we're kind of far away from it.
I can get closer. This is the maximum of the
Yeah,
camera
that's
right
a
now,
fairly large amphipod.
It certainly is. And Les Watling suggested that the amphipods, the organisms in the Okeanos were amphipods.
Watch leads, are you happy with that, or would you like any different views?
Yeah, that's the tightest we can get.
I could move closer if you want. But that's the.
We had a request for it, so it just a little bit farther. Is that a pycnogonid in the foreground?
Yeah, that could be a pycnogonid that's just on the bottom of our.
Look how slow he's inching along.
Right there?
Yes. That is a very slow one. So yeah, Pilot, if you could just inch forward a little without running over the corals we've been looking at.
Video compartment. Just gonna move around quick. I think Nav wrote it down.
You wrote a time down on that, Nav?
Yeah, it was 15:49.
Okay.
All right, video, you want to come back in?
That's great.
Copy. Well, actually, is that better?
That looks great. I'm waiting to see if I have any comments from the chat room. If they want more, that's as close as we can get without moving the ROV, but we could still do that. And I don't know if that's a whip coral. In the foreground getting in the way, but now we're seeing a little motion.
That looks more like an amphipod mud tube than a whip coral. So it's some kind of tube. There are some amphipods that build these tubes that they then just clamber up on.
No kidding.
And that's, I guess, there.
Yeah, I'm going to need help from the biologist right now because my 2 biologists just abandoned me and they went to lunch. Imagine that. So, Pilot, I guess that's a good view. And let's head on.
All right. Video, go ahead, come wide. All right, so a little push forward, get some ground.
We haven't seen that much variation in what the habitats are here. We're seeing lots of brittle stars, these grazing paths from heart urchins, several different urchins, pincushion, some people call them, but pancake urchins as well as the regular urchins. And up on the upper left, we see maybe our first outcrop of rock. Finally. So we're seeing some hard substrate, and automatically you start seeing more organisms populating the area.
Yeah, watch it. If anything jumps out that you'd like a zoom, just let me know.
Yeah, I think since this is our first look, let's zoom in on that bright white near the lasers right now.
Yeah, copy that.
And of course, as soon as the biologists leave the watch lead, we encounter this.
All right, video, let's go for zoom.
Looks like sponges from a distance, maybe lithisted sponges, rock sponges. And
interesting, yeah.
Leslie, since we're seeing some rock, we might want to consider a rock collection.
This is a great idea.
Yeah,
Perhaps
this
one.
A sponge may be related to what's referred to as a polymastia sponge.
Okay.
I'm not 100% certain of that, but it's definitely a sponge.
All right, and you see how dark these rocks are. We've seen rocks like this in the Charleston Bump area. This might be a manganese phosphorite type of rock. You can see a nodule-looking piece on the left.
I don't believe so.
Okay, I thought I heard you say
I think it would be. This would be a great sample. Let's look
might
first at the other rock pilot.
Spin and line up with you. Are you good like this?
Yeah. Do you want to center that outcrop that I'm?
Yep.
It's right in front of me.
How's that?
That looks great.
Awesome.
Okay.
Washley, is this a collection item?
Let's zoom out first, please, and because there was another rock with another organism just on the lower part of the screen. It'll come into view.
Copy
And
that.
maybe our folks online can tell us which they prefer.
Yeah, that to the left?
Yes, just to the left of the lasers.
Copy that.
With a little squat lobster hiding under the ledge. So you're seeing the ledge appearance. Yeah, these are interesting rocks.
All right, video, go for zoom.
Is that a tunicate or a sponge, perhaps?
Sorry, video might get a little dust cloud.
Scott, can you give me a potential ID of this?
Sorry, Leslie. I was chatting here in the lab.
Okay. Any idea what this is?
It looks like we're looking at a dead glass sponge, and I'm suggesting it's dead because of all the sediments that are on the outside. It seems to be
hasn't cleaned itself.
A bit more brown-gray than bright white.
Uh-huh.
But it was a glass sponge at some point.
Okay. So in terms of rock collections and specimen collections, we should probably go to the other sponge that you mentioned.
When you just
the one we just saw previously.
Hold on one second.
I wasn't suggesting a sponge for a collection. I was just noting that since we do see some rock, you might want to take advantage if there's anything loose and get a rock.
I didn't notice because I was looking at the camera.
Gotcha. Yeah, let's go back to the other one at pilot, if you don't mind.
Yeah, copy that. Video
and maybe set up for collection.
Copy. You can come wide then, video, if you're going to collect.
Nice video there of our squat lobster.
And Leslie, I certainly defer to you with the geology collection. I only say it because one of the sort of, I'll call it a protocol. It's not really a protocol, but one of the things we've been doing is on a dive, we try to get the deepest rock and then something near the end.
I'm all for getting a rock sample here.
That's why I suggested it. But if this doesn't look like it's interesting, then don't bother.
No, it's interesting.
So something to think about with this rock sample, guys, is...
Don't ask a geologist if the rock is interesting.
I know.
So something to think about, guys, with this sample is that if you wanted to collect a rock, we would typically collect a rock that doesn't have biota on it and keep it in the rock box. If it has something like this sponge on it, that's a little bit of a fragile biological specimen, you may want to put it in the bio box. And if that's the case, the pilots need to consider whether it will fit or not before they can collect it. So if you want to go for this rock, you probably want to put it in the bio box. If you want to pick as large of a sponge, then you can still get some cryptofauna associates on it.
Okay.
Just something to think about.
Is there anyone online who would really like that sample? Otherwise, it might just be easier to collect another sample of rock that won't go, or just put it in the bio, instead of keeping this one in the bio box, just put it in the regular.
Well, I can't tell you if this is a sponge that is particularly novel. I'm not convinced that rock is loose. It looks like it's just exposed bottom.
It might be.
Other consideration.
Okay.
So, Watchley, do we know what we want to do?
Let's, if you can zoom out a little bit, maybe there's an easier rock sample, unless anyone specifically wants this specimen in the bio box.
Video go, come wide.
There might be just a piece.
Sorry, that was wide, right, video?
Oh, that's it? Okay.
I can back out.
Yeah, because there seemed to be a little more rubble in the foreground.
Copy that.
And there's also a sponge in the well.
I'll just get a little height.
Okay.
And then there seems to be a lot in this general area, though.
There's definitely a lot in this area. So let's not go too far up. We want to get at the base.
Copy that. I'll stay low.
Yeah, so maybe right in the bottom of the screen, just to the left of, maybe, is that sponge? There's some loose, it might be some loose rocks, just above where you had landed.
In case you don't see the chat room, apparently a note from Scott Harris, it would be better to move up the mound to better rubble. Or he's asking would it be better to go to where there's more rubble.
Copy that. I can use the arm and poke some.
Scott Harris, should we were looking at getting something at the first outcrop, which would be this, the lowest. And then we would hopefully collect some more higher up. And can we zoom in on that sponge as well to see if it's maybe that rock next to this?
We do have a note here now that the polymastia sponge that was on that rock is not particularly exciting. So
okay, great, thanks.
Collection target. Thank you, Joanna, for that note.
Well, if right below where the shrimp is, in the middle of the screen, if that piece next to the sponge is loose, that might be a good small specimen.
Copy that. And then do you want that in a rock box or a bio box?
In the rock box. Yeah, in the smaller rock box.
We're not moving, are we?
All right. I will set up and I'll poke it.
And if that doesn't work, maybe some of the pieces to the left.
And do we have any samples?
Will work.
None.
Yeah. 0 samples. You have a drawer preference?
It's going to be a rock, so we're going to do...
oh, you picked up that rock.
Yeah, behind the sponge. Okay. So we'll just go port rock box.
So is this our first specimen collection of the cruise?
You want to get hydraulics up?
There you are ready.
It absolutely is.
That's exciting.
It is.
Yep, fully ready to go, and I believe you've already been bumped in. So would you like me to bring in the wing, or...
let me poke it real quick and then go from there.
So if you have just joined us, we are at the first rock exposure on our dive. We have traversed 100 to 200 meters across very flat sediment plain, but we saw lots of organisms en route. And now the slope is starting to increase here at the base of a low-sloped scarp, but we came here because Peter Etnoyer and his group wanted to investigate coral and sponge communities, and we picked this site because of its increased slope and that it showed a higher intensity return on the backscatter. And that, you'll hear us talk about backscatter periodically because that we kind of like to go to areas that have high backscatter. So
well, actually, that rock was loose. Do you want to continue collecting it?
Yeah, let's do that.
Thank
you.
So yeah,
great.
Go bring that in.
Oh, you ready?
Yeah.
Okay, Fort Wayne coming in.
So what we mean when we say high backscatter is in the collection of
you can put
water depths, the bathymetry.
We
use a multi-beam system, and we at the same time we're measuring the time it takes for sound to bounce off the seafloor, and that gives us the depth range. We also are recording how much of the signal is returned. We know how much leaves the ship from the sonar head and how much returns to the receiver. And we've learned over lots of years of study that the high intensity return, or as you might think of it as a strong reflection, typically is coming off of harder bottom or harder substrate. And in these areas, that would indicate to us rock exposure. If we are covering areas that are purely sediment, they tend to have lower intensity return. There's a little more scattering and absorption by the substrate. So we have pinpointed our dive sites seeking areas of higher slope, and ideally up to slopes of, greater than 25%, but we haven't found those kinds of slopes in this area. But this is steeper than the surrounding sediment areas. So we're looking for higher slope.
Piece of mud.
Did we get
it? Yeah, that was just mud.
It was. Well,
crumbled.
That's okay.
Sorry, Washley. Is there anything else?
So the slab that had the dead sponge on it that we were looking at earlier, that is clearly rock, and it's broken off. It's at a different angle. It's large, but it would be very indicative of these layers if we could get that.
We can move back and we can see if it'll fit.
Yeah.
Does that work?
Yeah.
Great. Let me just...
It might be ambitious, but I'm new at this.
Arms off.
Before you go, before you move, can you try the piece that is now right next to the sponge? I don't know if that rolled off or if it's the same, but it's very dark.
Just
to the
left.
right. No, to the right of the lasers.
To the right of the lasers? Yes.
Is that a piece that fell off as you pulled it up? Maybe if we zoom in just to look at it for a minute.
Yeah, video, you want to do a quick zoom? Video, you want to do a quick zoom? Yeah, quick zoom. Just go a little. Yeah, right, that's good video. So right to the right there.
Yeah, and next to that sponge.
All right, yeah.
If we can get that, that would be fine. If not, I'd like to try the larger rock.
All right, video, go ahead, come wide.
And you can see from that rock on the left of the screen, it's very flat and shiny, and that shininess may be from ferromanganese oxides precipitating out of the seawater and coating the sediment layers that existed at the time these were deposited, and I suspect that's why the rock on the far left with that sponge, ooh, that looks good. Oh no!
And
yep. So these are just coatings. That's interesting to see. These rocks are slightly consolidated sediments that have a ferromanganese coating on them. I believe that's why they're exposed this way. But let's try, Pilot, if we could try the one on the left. That'll probably break into pieces too, but we might get a piece of it.
yeah, copy that. We'll just reposition quickly.
Thank you.
And we'll try it.
So Leslie, while they're repositioning, why do we collect rock samples? What do they tell us?
Well, in our region, the rocks here are not volcanic. So if any of you out there have been watching the Pacific dives, that was a lot more lava and volcanic material. But here we have sedimentary rocks, and these are rocks that are the result of long periods of deposition from the seawater or from the continent, where material has come down off the continental margin onto the slope and settled out, and or from organisms that live above this area in the sea surface areas, and when they die, their shells settled to the sea floor. And when we zoom into any of these sediments, you often see the shell remains of many organisms. Over long periods of time, those layers of sediments build up and they get buried.
30%.
And burial then squeezes out water between the grains.
We need to watch them because they went down from like 36,
then precipitates. You get mineral matter precipitating out of that remaining water that helps to glue those grains together.
At what point?
And so when they get buried,
Jeff said 12 to 14 is empty.
To great depths, they actually get solidified into true rock. Otherwise, they're consolidated sediments, or we call it indurated sediments if they are still, they look like rocks, but they can still be disaggregated. They are considered indurated. And these, I don't know what I would call these kind of disintegrated completely.
All right, why don't you take the minis?
But,
all right, watch lead. This is Pilot.
Yes,
Back
yes.
with you. We just had a watch change.
Okay.
So as far as I understand, you want to try to go for the rock and dead sponge at the top.
Rock and dead sponge, yes.
Top left, is that correct?
Yes.
Okay.
And I'm just sorry it's not a live sponge, but Nolan Bear will be happy. We'll get a better look at the base of this sponge. He requested
and the ship's not moving and all settled out now, I assume?
Correct.
Okay.
We're not moving at all.
Okay.
So my guess is that this rock will actually crumble. So if we can grab it above where the sponge is, we might still get a hold of the lower part while it crumbles above. I don't know. But the two rocks we've tried to get have both crumbled in the grip of the manipulator arm. And we're going to have one unhappy squat lobster. So the rocks we're seeing are layers of sediments that had been buried, and so the consolidation, or what we call the lithification process, had certainly begun. The water had been squeezed out between the grains, and they started to cement together a little bit maybe, but they certainly had not fully reached a rock stage.
Right, we'll give it a poke.
So, I mean, we call them rocks, but they're not fully lithified.
It sure is a little wet.
It's rainy out there.
Okay. Force feedback is on. Pots all look stable.
All right. Hydraulics up. Our hydraulics up.
Yes, you're ready to go. All right.
So we're going to be attempting to collect the rock that has this